imagine life 300 years ago, long before your iPhone, long before the car, long before even the modern scientific method. I want you to really place yourself in that position and think about what life would have been like back then. Now imagine I told you way back then that our world was filled with microscopic life, tiny invisible creatures. Imagine I told you that these creatures were floating around in the air and swimming in your breakfast cereal and crawling over you as you listen to this talk. I feel like you might understand where this thought experiment is going. You see, 300 years ago, long before modern science, without any help of modern biology, even the idea of microscopic life would have been unthinkable. You would have called my theory strange, and no one would have blamed you for it because we didn't have the evidence to prove it. Well, my friends, I want to share that our reality is precisely this strange. Perhaps even stranger, and hiding in this strangeness are some of the most exciting mysteries that science has to offer. I'm Aryan Harshit. I'm an undergrad at Stanford University studying bioengineering. By day, I work studying microbiology at the Boyke Lab in Stanford Medicine. But on nights and weekends, I have a much cooler job title, microbe hunter. Let me break down what that means. A microbe at its core is any life or piece of life that exists at the microscopic scale. That is micron, or a millionth of a meter. That's about 100th the thickness of a human hair. And this includes all kinds of life, including bacteria, fungi, parasites, and essentially any biology that exists below the level of the human eye. Now, I see this talk as a defense of microbiology, because as a microbe hunter, I spend my time discovering new microbes, but I don't think the world entirely understands why this is so important and critical to science. So I want to give you an honest glimpse of this field from the inside, and I want to show you what makes this part of science so beautiful and interesting. So before we get into the details, I want to address a more basic question. Why would anyone possibly want to look for microbes? Well, the first and most obvious reason is that microbes are a fundamental part of us. Our current estimates suggest that the average human is made of anywhere between 30 to 50 trillion cells. That's a lot of cells. But how many microbes are in the average human? The answer is about 300 trillion. So that alone is a microbe to human ratio of about 10 to 1. So intuition alone should be telling us that microbes do something important inside of us. In fact, we found microbes in every major organ system in the human body. And we've shown that disrupting these communities of microbes, either with antibiotics or by selectively knocking out strains of bacteria, we can cause some serious long-range effects. Back in 2018, Alexander Vincent's group at McMaster University showed that mice raised in germ-free environments, that is, environments without bacteria, showed abnormal colon structures in their large intestine, compared to mice that were grown in standard microbe-filled environments. And so, these mice not only showed a difference in their structure, but also a difference in their ability to absorb nutrients from their food. This shows that microbes not only affect the structure of how organisms develop, but also their function. And even beyond the gut, we've shown that microbes are linked with several physiological functions, from our absorption of vitamins to the regulation of our hormones, all the way to the communication between parts of our body and the brain. Over the course of human evolution, microbes have literally stitched themselves into human biology. And so any attempt to discover the microbial world is also an attempt to discover ourselves. Now, the medical value of microbes is just the first layer. If you look carefully at the history of science, you'll find microbes everywhere, often in unexpected places. Back in 2008, scientists at Danisco, the milk company, had a problem. They realized that one species of bacteria in their yogurt, S. thermophilus, was constantly being infected by viruses, which actually affected the efficiency of how well they could produce yogurt. But when scientists tried to dissect what was going on, they found something fascinating. Somehow, S. thermophilus was physically storing snippets of DNA from viruses that had infected it before. And then when an identical or similar virus revisited again, a pair of enzymes would then match that DNA and snip it up. This was essentially a bacterial immune system. And this chance discovery from yogurt led to the discovery of CRISPR, which won the 2020 Nobel Prize in Chemistry and led to the basis of modern gene editing. Of course, the list continues. Microbes have already enabled things like penicillin, the PCR tests that we relied on during the pandemic, blood thinners like warfarin, and of course, vaccines. And there's a reason microbes are so influential in both scientific and biological progress. Over the past 3.5 billion years, microbes have braved some of the harshest conditions and competition on the planet. Here's a little brief snapshot of this. In the past few decades alone, microbiologists have isolated viruses from the Marianas Trench, where the average pressure on a human would exceed 100 adult elephants standing on you. We've isolated fungi from Antarctica that can survive in space-like vacuums and lethal doses of cosmic radiation. And in the bacterial world, we found C. metallodurans, which is capable of naturally picking out copper and gold from its environment, and is actually being explored as a tool to make mining more sustainable. Now, these are just a handful of interesting microbes that the world has to offer to us. But it begs another question: how much microbes have we actually mapped? If we take viruses alone as a case study, some of our largest databases say that we have about 70,000 viral genomes cataloged. That seems like a lot, until you wonder how many viruses are actually out there. Current estimates suggest about 100 million unique species. And this is a number from Dr. Vincent Raffagnello at Columbia University, who did pioneering research on the polio virus. Now, if we think of viruses as a good model for microbes as a whole, it suggests something pretty shocking. We've discovered maybe 0.1% of the microbes on our planet, which should make you wonder, if we've discovered so much influential and important science with just 0.1% of the microbes out there, what else is hiding in that other 99.9%? I think we have every reason to believe that there's mountains of obvious and amazing progress hiding just on this other side of what we discovered. Now, we've established that microbes are important in many ways, and that most of them are still unknown to us, but how do we actually go about filling this gap? How do we discover new microbes? Now, I don't want to oversimplify, and there's so many methods to do this, but if you were to distill the principles of discovering microbes into a few basic recipes, a few essential mechanisms, you'd arrive at something like this. Microbe hunters first explore nature, collecting samples of places where their microbes would naturally thrive. From there, they select microbes that they're interested in by selecting for certain biological traits. Finally, after purifying microbes, they can study them all the way from the genomic level, so DNA and RNA, right up to their structure and biological function. As a real example, I study a microbe called Pseudomonas aeruginosa. It's a green rod-shaped bacteria that's responsible for thousands of skin and lung infections every year. Back at Stanford Medicine, we see Pseudomonas taking lives every month. And since many strains of Pseudomonas are actually resistant to antibiotics, this remains a giant unsolved problem. So in my research last year, I was interested in looking for viruses in the natural world that could safely and effectively destroy Pseudomonas. So following the microbe hunter's playbook, I started by snapping on my gloves and exploring the natural world. You see, since viruses that infect Pseudomonas and viruses in general are parasites, you won't find them very far from the cells that they infect. And so in order to find viruses that infect Pseudomonas, the best place to start is by looking for places where Pseudomonas naturally thrives. And in this case, that would be things like soil. I even swabbed random areas of my lab, and eventually even emailed Stanford's wastewater facility, which essentially provided me with this giant slimy vial of bacteria that I used for my experiments. There's a problem here. Whenever you extract bacteria from nature, or any microbes from nature, they live in these complex communities full of bacteria, virus, fungi, and everything in between. So how do we take this mess
and select for the viruses that we're interested in. The first step is filtration. So we can run our sample of soil or wastewater through a polyether sulfone filter, also known as a PES filter. What makes PES special is that it's engineered with pores that are about 0.2 microns across. That's about 0.2 millionths of a meter across. And what this means is that bacteria and fungi, so large microbes, can't pass through, but those holes are just about small enough for viruses to squeeze through. From here, to select the viruses that we're interested in, we can use the right cell as bait. So in my case, this was a strain of Pseudomonas. Essentially, when we introduce Pseudomonas to this filtered virus, all the viruses that can recognize Pseudomonas multiply like crazy, and they overshadow any other microbes in the solution. But viruses that don't stay stagnant. And so through this method, we can arrive at a collection of viruses that almost exclusively infects Pseudomonas. When we incubate these viruses with Pseudomonas, we get something like this. Those clear circular zones you see are regions where Pseudomonas viruses have completely destroyed that bacteria. We call those zones plaques, and different viruses show different shapes and sizes of plaques, which can help us identify them. And so, if we repeat this process over and over again, we can keep extracting viruses until all of the plaques on this plate look the same, which means we have a nearly pure sample. After I arrived at what I thought was a set of new viruses, I wanted to poke around and understand how they worked. For most microbiologists, this starts with a genome. After sequencing the DNA of these viruses, I use special bioinformatics software to generate these structures called phylogenetic trees. You can think of a phylogenetic tree as the microbial version of a family tree. So viruses that are genetically close to each other are grouped close, and distant relatives are grouped further apart. And using this approach, I proved that 2 of the viruses I discovered counted as new species, which were actually proved internationally just a few months ago. From here, I wanted to test how these viruses performed in a real-life setting. What you see over here is a plate of Pseudomonas that we extracted from a real patient at Stanford Medicine with a drug-resistant lung infection. And all those plaques you see are one of the viruses that I extracted called Vanta. Within the course of 24 hours, Vanta cleared this entire plate of bacteria. This was a virus discovered in nature that's capable of destroying bacteria that human-made antibiotics could not. My hope is that viruses like these will become our next generation of treatments against bacterial infections. In fact, approaches like these are already being used in Eastern Europe and are already in the clinic. And they're going to be in the clinic soon in America within the next few short years. So to return to our original question, why is studying microbes meaningful and why should we care even if we aren't microbiologists? Well, I'm a big fan of poetry, and I think Walt Whitman said this really well when he wrote, "All truths wait in all things." I think if there's anything we can learn from microbes, it's that biology is deeply interconnected. And so any attempt we make to study the world of microbes is inevitably a journey into ourselves. And I think this reveals something deeper about how humans make sense of the world. As humans, we tend to believe what we see, and we tend to see what we want to believe. It's our nature to ignore what is invisible to us. For most of history, biologists thought of life as flat map. We thought the world that we could see was the entire world, and that everything else was just noise. When in reality, microbes forced us to see something different. We learned through microbiology that most of the world, most of life, was actually invisible. And so yes, biology might be a single map, but it's also a million maps at a million different scales that we can't even see. So all this time, we've been staring at a single slice of life, a single scale of reality. And if you talk to any microbiologist today, you can sense their excitement about the moment we're in, because it's almost like we've unlocked an entirely new library of universes, and we get to be some of the first people who study this. So if I told you 300 years ago that our world was full of microscopic life, perhaps you wouldn't believe me, and you'd have reason not to believe me. 300 years later, I'm here to tell you that reality has gotten far, far more interesting. life's biggest frontier might actually be our smallest frontier at the same time, and it's wide open for curiosity. To align with this, some of life's most important questions trace back to microbes, and the answers to those questions inevitably trace back to us.